to Science, Episode 5.
Well, let's give this some context. We are here at UNCG with John Tomkiel Dean, who has been kind enough to speak with us today about his genetic research and what he does at UNCG, and fruit flies, so, to put it very plainly. But if you want to elaborate on just a little brief.
Some of the stuff we've been looking at.
Yeah.
Oh, sure. Yeah. So flies, a lot of fly researchers think of flies as little humans with wings. And we try to sell that to get grant money. But they're actually really remarkable in how similar they are to us. About 65% of the genes that we know cause human disease are conserved in flies. So we can study those disease-causing genes in flies and manipulate them in ways we can not manipulate humans. For example, we can do forced matings and dissect the offspring, and things that people kind of frown upon when you do in humans.
Just a little bit, yeah.
So we're interested in a particular problem called non-disjunction, which happens when you're making eggs and sperm, where chromosomes misbehave, and instead of producing an egg with the proper number of chromosomes, you're missing one or you get an extra one. And that's a huge problem in human biology, actually more so than any other organism that we've studied. If we look at a model system like the mouse, they make errors at somewhere around the rate of 1 in 500, 1 in 2,000. Every time you produce an egg or a sperm, 1 out of 2,000 might have an error.
Okay.
And in humans, in particular human females that are approaching the age of 40 or so, that error rate goes up to 1 in 10.
Wow.
So some of that's due to the particular aspects of human biology that differ from animals like mice. We live a long time. So human females carry their eggs around a long time, and they're exposed to ionizing radiation from outer space, UV light from the sun, chemicals in the cafeteria food, all of those good things. And those can break down the eggs over time and affect how the cells divide. So part of that problem is looking at environmental causes, looking at things in our environment, things like BPA, bisphenol A, which you probably heard about, is the plasticizer in water bottles that is somewhat eliminated from plastics now, but is still in liners of vegetable cans and is still abundant on cashier receipts you get.
Cashier receipts, really?
The ones, the heat-sensitive paper that fades with time.
Yeah.
The thermal ink paper.
The thermal ink
wow,
paper, yeah.
I had no idea.
BPA, and it absorbs through your skin. So you're getting a good dose from common reagents in the environment still. So there's a huge area of study of looking at those environmental causes, but we're more interested in the biological causes, the genetic causes.
So you aren't rubbing BPA on flies?
No.
Okay.
What genes control how chromosomes move, and then what goes wrong with those genes to perturb the production of eggs and sperm. So in humans, this is both sociologically and economically a very important problem, because still amongst live births, about 1 in 500 have chromosomal problems. These are whole chromosome errors where you have a whole extra chromosome or missing one. So these are trisomy 1 children with Down syndrome, or Klinefelter syndrome, which is a feminized male, or Turner syndrome, which is a female who's missing an X chromosome. So they're very, very common relative to any other genetic disease you can think of. If you look at something that's really frequent, like cystic fibrosis, that only affects 1 out of 20,000 people or so. That's a very, very common genetic disease. We're talking about 1 in 500. So this is a huge source of genetic syndromes in the human population. We don't understand really much about how we can make that better. So if we understand the mechanisms, maybe that will give us some idea of what's going wrong, some idea that we haven't thought about yet.
So is there any prevailing theories right now that anyone's promoting?
Well, with the human problem and this relationship with maternal age, there are some theories. There are some proteins called cohesins. Let me show you. So when you replicate your chromosomes, They are bound together
Right.
by these ring structures called cohesins, and if those break down, the chromosomes will prematurely separate,
Okay.
and that can cause errors. So people have shown that cohesins actually break down with age and become become less abundant in eggs of older females, and that may lead to precocious chromosome separation, and that's a cause of this non-disjunction.
So it just breaks down over time, or is it
Over
like
time, yeah.
Okay.
So, and that could be influenced by environmental factors like chemicals or pesticides, things that might exacerbate that.
Okay. So you don't have an actual treatment for that, or is that just kind of, has anyone found a way to strengthen those, or?
Well, if you can find that relationship, you can try to reduce the exposure.
Okay.
So that's one way of thinking of the problem. But we're actually more interested in just how the whole system works.
Okay.
So the most interesting part about making eggs and sperm, this division we call meiosis, is how chromosomes recognize each other, because in the process you have an identical chromosome that mom gave you and dad gave you, and they have to find each other, pair with each other. And that's a process we understand very little about, and that's the reason I turn to the fruit fly, and the male fruit fly in particular, to try to understand that process. So flies are interesting in a number of ways. They're not only a good model for learning about how basic biology works in humans, but they've evolved completely different mechanisms of performing this division of meiosis, and we don't know why. So from an evolutionary perspective, it's interesting to find out why or how these different systems evolved, and what are the different variations you can have on this basic mechanism. All diploid organisms that get a set of chromosomes from mom and dad, they all do meiosis, but flies do it in a variety of ways that differ from other organisms.
Interesting. And that doesn't change how they how they replicate for study purposes in humans, even though it's different.
Well, some of it won't be relevant to humans because of those differences, and some will. So understanding what's shared and how that differs is sometimes interesting as well. In most organisms, when chromosomes find their partner, it's like walking into a bar and trying to find your partner, right? The bar is full of people, and you're looking for someone similar to you. So you have all these interactions with other people in the bar, and there may be some similarities which might cause you to linger with that person, and then you find out they're really quite different from you. They support Trump or whatever, so you move on to until you find that exact match, and then that's stabilized by an exchange of DNA, which chromosomes actually do a process of crossing over, which not only mixes up the DNA, but it ties them together so that they are divided properly.
Okay.
And so male fruit flies don't do that. They have the same pairing process. They have to find their partner chromosomes, but then they don't exchange DNA. So we don't know why, because that exchange of DNA makes new genetic combinations, and evolutionarily that's a beneficial thing. Your offspring could survive new environments if they have more variety, more diversity. So female flies do it, though. Female flies are much more like humans. They have this crossing over, this exchange of DNA between their paired chromosomes, and males don't. So not only was it interesting to find out, use this as a system for pairing, because in some aspects it's a simple system. They pair and then they segregate. Where other systems pair, they do this crossing over recombination and then they segregate. So males lack that complex step, so we thought they'd be a simple system. And then it's kind of an evolutionarily interesting question. Why do they do that? Why does it happen only in one sex? Why are males different from females in that respect?
Yeah, I'm just trying to think of how. So how do they share the DNA? How does the offspring take the DNA?
Well, so in males, for example, you have an X and Y that are partner chromosomes. So they pair, and then at the first division of meiosis, they move apart to separate chromosomes. So there will be X-bearing sperm and a Y-bearing sperm. In a female, two X's would pair. They would recombine and exchange segments of DNA, which holds them together to the proper time. Then they segregate.
Okay.
So males are missing that step of recombining, and it's hard to imagine why that would be advantageous.
Yeah.
So we've been looking for genes that control that, and the first one we found is actually in that process of holding them together. So in flies, there's a history of naming genes for the defect they cause.
Okay.
So in this particular mutant, the chromosomes come together, they pair, but then they fall apart. They can't hold on to each other. They can't stick to each other. So we call that gene Teflon. And Teflon is one of the alternative proteins that holds paired chromosomes together until it's time for them to separate. And it's only evolved in flies. You don't find it in any other organism, but you find it in all dipterans, or two-winged flies. And there are multiple genes just like Teflon.
Okay.
M&M and S&M or some other part of this complex that hold chromosomes together.
So does that result in much greater genetic diversity among female flies as opposed to male flies?
No, so the recombination rates, the rate at which they do this exchange of DNA is about the same in flies as every other organism.
Okay.
It's typically one per chromosome arm, one exchange per chromosome arms. So females have not increased their rate of exchange to compensate for the absence in males. Males have just gotten rid of it and evolved this whole new system to hold their chromosomes together. So when we further studied Teflon, we found that it only holds some chromosomes together. The sex chromosomes are unaffected by Teflon. So actually males have two systems. They've evolved independently to hold chromosomes together without recombination. So it's a mystery of how these evolved, where they came from, what their general purpose was.
And has that led to any discoveries or any kind of anything?
We're hoping in the future it will lead to some more interesting. So we're still trying to backtrack from that. So that's the step after pairing occurs, binding those paired chromosomes together. What's really interesting is how the chromosomes pair and find their partner in the first place.
Okay.
So we're hoping we can maybe backstrap from that gene, find how that assembles on the chromosomes, if that's involved in the actual pairing process, and looking at pairing itself.
Okay.
You said something I always find interesting when scientists use the word we.
We the field.
We the field. And I think that is a really fascinating way of looking at things, because some people in corporate or in, some scenario where there is, not open research and not this field of people trying to achieve the same goal, that doesn't necessarily exist. And
well, that's not to say that there's not competition in science.
Sure. Yes, that's
and,
true.
There are labs that work on very similar projects that we do, and we share our information through publications, but we keep a little to ourselves until we're ready to publish. There's always a concern, and that's been driven by the tightness in funding.
Right.
Right. Okay. So the entire NIH budget, is less money a year than we've spent on B-2 bombers.
Right.
So.
And is that a pronounced decrease in recent times, or has it always been less than?
It was pretty static for a long time, and then there was a doubling fairly recently in Clinton's time. So it's up to about $35 billion a year.
Okay.
But that's funding the majority of medically related research at all the universities across the country. When you start to break it down, it's really not that much money.
Right.
If you start to put it in context of other things we buy,
right.
It makes it very competitive to get government funding to do research. So probably about the top 15% of grants are funded. And you can't simply keep throwing in more grants and buying more lottery tickets. Writing a grant requires a lot of preliminary data, years of work sometimes to prepare, to get at the right point to ask for funding. So it makes it a very difficult environment to work in.
Sure.
So that is the university investing up to the point where the grant can even be applied to? Or who's covering, the preliminary research just to get to grant?
So universities try to do that. There are some slush funds and some state funds. In North Carolina, there's a North Carolina biotechnology fund that will give you some money if your research can be translated into some biotech use. But covering basic research is the most difficult, and that's where it's been hit the hardest. Where you're doing research without, for example, studying some weird system of meiosis in fruit flies, where you can't imagine what you might discover.
Yeah.
So recently, somebody was studying some weird system of finding duplications of DNA in bacterial genomes, and that turned into a new way to edit all genomes. This CRISPR-Cas9 system came out of very basic research, studying how bacteria defended themselves against viral infections. No one could have imagined that would be a way of replacing genes in humans.
Right.
The discovery of RNAi, which is a way to regulate genes, was very similar. Somebody was studying a developmentally regulated gene in the soil earth nematode, C. elegans. And they found it was regulated by this RNA mechanism, and then we can use that mechanism to regulate genes in humans as well, in cancer therapy and different treating different diseases. So you never know what discoveries will come out of basic research, and they're not necessarily related at all to what you're looking at.
But you find it is hard to justify to funders that you want to just do research about something that might not have an end game.
For the purpose of research, for the purpose of new knowledge, it's hard to justify that.
Which is sad, I think. That's really...
It is, so it turns the grant writing into how you can best justify this for human health.
Mm-hmm.
And it turns it into an environment where what gets funded are the obvious next steps. So the imaginative work, the stuff outside the box, the bizarre systems, the places where you might lead to really new discoveries don't get funded as often.
Are there places in the world that do better at funding things like that? I mean, internationally
than the United States?
Yeah.
I don't think so. I think we're probably still the leaders, although China is investing a huge amount of resources. We train some of the best Chinese students in the world, and for the most part, they stay here and are running the research labs here and are really contributing to our system, but I think that will change.
I read recently that China is using CRISPR to treat human cancer patients, and they're actually applying human trials now, which I think the U.S. is still several years away from that.
Yeah, so that may or may not be a good thing.
Exactly, that may or may not
how
be a
you
good
view
thing.
experimenting with human lives. We tend to over-regulate a bit, but I should say err on the side of caution, where other societies don't so much.
Sure. The whole anti-Hippocratic
At least we don't give government funding to do that sort of high-risk behavior. The first studies on CRISPR in human embryos were done in China to show, it answered a very important question. We use this technique to modify a particular gene, but it has what's called off-target effects, where it may hit the wrong gene and make a change there, which could lead to cancer or some undesired outcome. So you have to know how good your system is, how targeted it is, and how to reduce those off-targeting. And these Chinese researchers were able to show that there are significant off-targeting events by doing it in human embryos. You need to do that experiment to know how it works in human embryos. You could do it in the fly, but that still doesn't tell you how it behaves in humans. But
what do you think of the potential for computer models to eventually be able to be created that synthesize the human genome in a way that
that's really exciting.
So
there's the big problem with that for years has been not predicting so much how genes work. I think we're getting closer to looking at gene regulation, how that's controlled and manipulating that. For example, we can create stem cells by expressing a combination of 3 different genes in those cells now and causing them to revert to a stem cell behavior, which will allow us to develop those into different lineages, like neuron replacement or growing them into kidneys or livers eventually. But proteins, the end product of the gene, are really hard to predict their structure and their activity without studying each individual one and doing X-ray crystallography and really figuring out the nitty-gritties of how a protein is folded and functions. So it's not quite computerized yet, but I could see that happening down the road. There are many fewer genes than we ever expected to be in humans. Humans have about 35,000 genes. Flies have 18,000 genes, so a lot of plants have a lot more genes than we do. We're not as complicated genetically in terms of puzzle pieces as we thought, but we're much more complicated in how those pieces interact and work together than we imagined. So the big blossoming field now is the field of epigenetics, which is how environmental influences can change our genome or change the expression of our genome. Not the DNA we have, but how the DNA is used, how it's read. And there's some really striking studies of transgenerational effects. And one of the recent hires we made in the biology department is an expert in epigenetics. He looks at treating mice with pesticides and then following up to 5 generations later, in untreated generations later, higher incidences of cancers and different inherited diseases. So understanding how our genes respond to the environment and get changed in a way that we can actually pass on to our children is a big mystery. There's a lot of mystery still to be unraveled before we can stimulate it or simulate it all on a computer.
So is there anything coming up that you're looking forward to as far as some new research or something you're going to be doing here? Or something someone else is doing?
It seems like I just got a grant, but my grant's about halfway done now. But we've got an NIH grant to study pairing in the flies, and using CRISPR-Cas9 to make deletions of the genome and see how deleting different parts of chromosomes affect this pairing step. So I hope that will turn into something very interesting.
So you're studying like the accidental targeting with that also, and just seeing how accurate it can be?
No, in flies actually we don't really care, because if we're targeting chromosome A and there's a mistake made on chromosome B, we can cross a couple generations and replace that chromosome B.
Okay.
It's not something you can do with a human. So if you're born with a genetic error that's induced by some manipulation,
that's just a lifelong problem.
Yeah, it's a bigger problem than fruit fly. Fruit fly, you go into Fly Morgan.
Which, as we said, was what, 99% ethanol?
99, he said 80.
It's a little bit of benzene in there in the distillation process.
So, and you're just looking at how the CRISPR affects and how it passes on? Will the changes pass on?
No, we're just using it as a tool. In flies, we will delete segments of chromosomes and watch how those chromosomes behave in this pairing process. So it's already been, in just a matter of years, people have set up toolkits to use CRISPR in a variety of model organisms. Most of the systems already existent in flies, so there have been the enzyme that catalyzes the DNA changes has been placed into flies, and we just have to order it from a fly stock center. Here it comes, and we can target the genes we want using that system.
So do you actually, and I only understand the CRISPR stuff on a very high level, do you actually make it here, or?
We make part of it. So CRISPR involves an enzyme that will cut the DNA.
Okay.
And a small piece of RNA that will target that enzyme to tell where to cut. We make the small piece of RNA, or we pay a company to make it, or we make a gene that makes it and make a transgenic fly. And we cross it to a fly that has the enzyme, combine the two parts, and then the system will
So is it something you
changes we
feed
decide.
them, inject them with, or how
You
does
inject it into fly embryos, and you select for flies that have taken up the gene.
Okay. Interesting.
Yeah, so transgenic flies, it's kind of fun to make them.
Yeah.
We the last one we made was my master's thesis project. We were looking at a gene we thought was involved in this meiotic division, but it turned out to be involved afterwards. So we took the gene and put it in the fly, fused to a gene that makes a green fluorescent protein. It comes from a jellyfish.
Okay.
When you shine a UV light then on the fly, the protein that you make from the gene glows green. And instead of finding it in the meiotic divisions, we found it only in the mature sperm. So this was a protein that was loaded into sperm after sperm were already made, or in the process of making sperm. And then when that sperm delivered the chromosomes to the embryo lost chromosomes. So we're still looking at a chromosome movement issue, but it was not in the meiotic divisions we were hoping to study. But it's kind of fun looking at the flies and watching their green sperm swim around. This is an aside, but flies have amazing sperm.
Very, very active.
If you were a fly, your sperm would be the length of your legs.
Nice.
So it's, compared to humans, they're about 100 times larger. They have huge tails, but tiny heads. So the sperm looks just like a needle, like a thread with a pointed end.
So how many sperm do they make at a time compared to humans?
Not quite as many, but they transfer a couple hundred to a female in mating, and
Okay.
she stores those in an organism that looks like a donut, and they swim around in circles. She uses them one at a time. As she lays her eggs, she fertilizes the egg just before laying it.
Huh. So she takes on 100, just carries them around, and can deposit them one at a time into an egg.
As she lays the egg, yeah.
Interesting.
And the sperm have this enormous length because the mechanism of fertilization is different than humans. In humans, it's kind of an all-out attack. Millions of sperm surround an egg, and the first one that gets in closes the door and changes the membrane so no others can get in. In flies, the strategy is to have a tiny hole the width of a sperm. So one sperm is released, finds that hole, enters the egg, and as it's entering, it trails this huge tail behind it. So it takes a long time for the whole tail to enter. Should two sperm get there, only one can fit through the opening, and by the time the other one comes in, the egg has already been fertilized and has started developing into a fly.
Wow, So does the tail actually carry any information, or is the tail just a movement mechanism?
It's just mostly what we call microtubules. It's proteins that have motors on them that make a sinusoidal movement, and a mitochondria that runs the length of it that makes energy to power the movements.
Okay.
So we don't think the tail really has any purpose other than to block the hole and prevent another sperm from getting in.
Interesting.
They're really a beautiful system to look at, too. The sperm looks like a ram's horn, or the testis looks like a ram's horn, and you can follow the development of cells from the tip all the way down. So they're spatially and temporally arranged in
Okay.
order. The young cells are at the tip. As you go down, they get older and older, and you can see where the meiosis occurs, and then where sperm start developing and growing tails, and you can very easily see the whole thing. So it's a good thing a good system for students to look at too. It's very easy to see all the developmental stages, to see where things go wrong. One other gene we studied had a really striking developmental impact. It changed the shape of the nucleus, and for some reason that resulted in chromosome transmission defects. So it's very much like this human disease we were talking about a little while ago, progeria. It's a protein in the nuclear lamina, which is a structure that gives shape to the nucleus. In progeria, it's called lamin A. It's the main protein that does that. And if you perturb that, you get this rapid aging defect in humans. The protein we look at is called topoisomerase interacting arginine serine-rich protein, or TOPRs for short. And in flies, it causes a similar defect in the nucleus, but only in the male germline, so only sperm are affected.
Okay.
Disrupts the chromosome segregation. But in humans, that same gene is a tumor suppressor. So it's associated with tumors, stomach tumors and lung tumors. And so we're thinking that these changes to the nucleus that we're seeing in meiosis that regulate chromosome transmission may be doing something similar in human cancers. So that's sort of another example of how studying one thing has led to maybe a tool for finding out about something that's relevant to human disease.
Good.
Were you always a weird dude, or did you?
I was always. I always wanted to be some kind of scientist. I knew that from when I think 4 years old. So I didn't know anything about science. I had the geeky microscope and the chemistry kit and the geology kit. And I always liked gardening, though. And I can remember being probably only 5 or 6 and Bert would come out with this contest, $10,000